But yeah, let us know if you have any objections to the recording, but it's always nice to see some friendly faces, so feel free to keep your cameras on if you want. So I think we're just going to get started. Today we are pleased to invite Yordi, who is from the BETin Institute, who will be talking to us about biotechnology in Ethiopia. Following Yordi's talk, we'll have a few Q&As, see what bright ideas have been sparked, some conversations from her presentation. We're going to then follow up with some kind of updates about what's new and going on in the Reagent community from the organizational side, but also it'll be great to hear from community members as well. So if you have anything new that's come up in your research that you'd like to share with us, or just any updates you think the Reagent community will be interested in hearing about, feel free. We'll be having a little bit of an open discussion afterwards. So that's all from my end. I'm just going to pass you on to Yuzi, who will introduce our speaker Yordi today. Over to you, Yuzi.
So hi everyone, my name is Yuzi. I'm an undergraduate student from Imperial College London, and I'm also a member of a Recon community. So I'll introduce our speaker, Yordi. Yordi is an associate researcher at the Bio and Emerging Technology Institute in Addis, Ababa, Ethiopia, and is a team leader for enzyme technology focusing on industrial enzyme production. She received her master's degree in biotechnology from Addis Ababa, and as a group leader now, she works on production of enzymes from extremophile microorganisms for industrial applications, mainly using indigenous yeast strains for production of chemicals from industrial sugar byproducts. We'll hand you over to Yordi now for her to tell you a little bit more about biotechnology in Ethiopia.
Hello, everyone. As already explained, I'm Yordanos from Ethiopian Bio and Emerging Technology Institutes. It is a governmental institute working on research on biotechnology and is having a mandate on coordinating and leading researches related to biotechnology. So my talk is going to be biotechnology in Ethiopia, but not the overall look of what biotech in Ethiopia, specifically more on what we are working in our institute, and I will give you highlight on what other institutes on this area of biotechnology are working on. So my outline will look like this. We'll discuss about what's BETin and research activities in BETin and how the society responds to our research activities and our coordinating activities regarding to related to the biotech. The picture that you are seeing is a member of the institutes. These are researchers and support staff during the establishment of our institutes. So as BETin as research institute, having different mandates. The first mandate given for our institute is research. We work on high impact researches and that can deliver product and service for the society. What I mean is that in our research institutes, we're not doing kind of basic researches. Researches that are done in our institute should be have impact, and we have to produce a product, and that product have to give service for the society. The other is we give trainings, focused trainings, technical and short-term trainings for different institutes. That means for we give trainings for research institutes plus universities plus for high schools even. We give some kind of hands-on training on the molecular techniques, and the other role that's given for our institute is advisory role. It is a mandate given before a year for our institutes to support institutions and governmental for achievement of their research. So for doing this, the institutes have different research directorates to accomplish our goals. The directorates are listed here. We have plant biotechnology, animal, health, industrial, environmental, bioinformatics and genomics, nanotechnology, material science and engineering, computational intelligence, reverse engineering, technology incubation and commercialization directorates. These are specific for specific researches. My area is on the industrial biotechnology. I may say a lot about it, but the other directorates also work research related to this biotechnology specific to their topic. And the last that you are seeing, the technology incubation and commercialization directorate is responsible to we transfer the technologies or the research products that we are found from our research activities. So the technology incubation unit will see how we can transfer and commercialize it to the community, because and for private stakeholders, because our research institutes doesn't have a mandate to commercialize or to sell our products. So this unit will transfer the technology to the private sectors or any interested group to take on the products that we develop from our research. Major activities that we accomplished so far is the first thing is increase public awareness level about the biotechnology. Well, what I mean is that because the awareness related to the biotechnologies goes to the negative, because mostly when we discuss about biotech, the society perceives it as everything on the biotech is just GMO. So we need to work a lot on this to increase the awareness of the society, what we mean by biotech, how it's important for the society, how the society can use the products from these research activities. Maybe in your case, you may not face such a difficulties to explain what biotech means for the society or for the end users. But in our case, the major problem is the awareness on the biotech, because there is already believed that the biotechnology works are highly related to only to the GMOs. So the society perceives GMOs as something not good enough for Ethiopia. So we work a lot on this awareness question. We use different medias, magazines, any medias we find to explain to the society what we mean by biotech in the language that they can understand. Because the language we use for researchers and the language we can use for normal society have to be different, and we try a lot to explain in the language that they can understand what we mean by biotech. Even our directors, researchers, everyone is highly involved on this. These are some of the pictures that you're saying published on the magazines, and this is our director giving a public speech on what we mean by biotech. The other is molecular research activity with great achievements. What we mean by that is that we're trying our best to work a lot on this molecular research activities, because so far the country research centers are highly engaged on the molecular, only microbial research activities. So we trying to higher up the research activities to the molecular level. So up to sequencing, we're trying to go to up to sequencing, and we have a sequencer in our institute, and we're trying to start on sequencing of DNAs. The other is identification of potential microbes for production of enzymes. As I'm a team leader for the enzyme technology team, I can say a lot on this. We're as the country is rich on the biodiversity, we have the higher chance to get novel microbes for production of novel enzymes. So we are trying a lot on this, and we have some isolates for production of different enzymes, specifically for the industrial application, like protease, amylase, xylanase, and currently we are working on xylanase, and on the isolation of even yeasts for ethanol production for different alcohol beverage industries. And the other is we give hands-on training on molecular techniques because there is a big gap on the country research centers and higher institutes on the molecular techniques and practical activities, how to work on. Even if there is a gap on the reagents, still there is also a gap on the techniques, and we give hands-on trainings for different research institutes. We arrange different training on research and development capacity buildings. And finally, we support youth groups to open their startup companies. What we mean by that is that after having a product, we give trainings for youth groups or interested groups. It may be outside of the capital city, so they can scale up the product to open their own startup company in a small scale. The one that you're seeing in this picture is that groups of people or youth trained for production of, with the blue clothes, they are trained for the production of fertilizer. So we try to train people from different sectors. Here is just example researches that we are currently working on. The first is isolation, characterization, production of bacterial enzyme for various industrial applications. But the production in this case is on the small scale, because we're trying to scale up the product that we have in our lab, and we need a lot of collaboration on this area, because we isolate the strains. We know they are efficient on production of this protease, amylase, ablase, phytase, and xylanase that we are currently working on. But the scale up is a big problem for us, because private sectors are not that much interested on the scale up of these enzymes, because it takes much time for them and they have to invest on it. So we need a collaboration on this to scale up the production of this enzyme from the lab scale to the industrial level, or at least to the pilot scale. We can use these enzymes for different bakery, dairy, beverage, leather, textile. Every industries need the input from the enzymes. So we're working a lot on this, and we are looking for how we're going to scale up the production of enzymes. And we're trying to work on this with Jenny Beneficial Bio on the production of different enzymes from wild gene plus the recombinant ones. The other is cell cross optimization on ethanol tolerant yeast strains, because we have ethanol production in Ethiopia, but from the sugar industries produce ethanol from the substrate that they find from the molasses from after the sugar production, but they have the yeast strain that they are imported for 13 years from India, and the strains get minimize their activity year to year, so they request us to look for a strain which is active and competent enough to produce the kind of ethanol they need to produce. So we are successful on isolation and having the yeast strain that's capable to work for the environment that they are working for ethanol production. These are simply just for you to show exemplary researches. And the other is production of Taq polymerase at laboratory level in collaboration with Beneficial Bio. We're trying to produce lab reagents in our lab, even if we're not going further on this, but the area that we are highly working on, because the reagent costs are very expensive, especially to work on the molecular area. We can't cover the cost for importing this reagent, so we're trying to produce our own research reagents, at least for the lab use. So we get training from Beneficial Bio on the production of Taq, And we're still looking for different trainings and to scale up the production of this Taq polymerase and other reagents for molecular activities. And we try to duplicate the kind of training that we find from Beneficial Bio to other higher institutes, which is outside of Addis Ababa, the capital city of Ethiopia, one of the higher university in Ethiopia, which is Reagent University. So we duplicate similar researches there, and they were very interested on their training to produce their own Taq polymerase in their own lab, but still there is a gap on the finding the reagents needed for the production. But we're keen on such kind of trainings and capacity building to produce our own reagents, at least for research purpose. And we will need to say thank you for Beneficial Bio team for preparing such kind of training for us and enable to increase our capacity on the production. The other is that our institutes not only working on the higher research activities, I mean, not only on the molecular level, not only on the enzyme, but we try to go down to the society and we look for what the society is facing day and night. So this is the plan that you seeing here is saying known as INSET. It is main food source for more than 20 million Ethiopians. But the way they are processing for production of food or meal from this plant is very tedious, and it's a lot of, you have to invest a lot of energies for cooked food from this because they don't have a starter culture to ferment this food. So what we're trying to do is that to introduce potential lactic acid bacteria as a starter culture for them to enable them to make their own food within a short period of time with less energy. Not only the starter culture, but also we design the machinery. We have a material team in our research institute. So the material team designed the material that is capable of processing the food, and if they have the starter culture and if they have the machinery, then their life gets easy. So we try to work such kind of works to not only the higher level researches on the molecular level. This is a model research. The other is model research for production of this drink that you're seeing on the picture. We call it that. It is Ethiopian traditional drink, and it takes much time for production. So we need to standardize the production process and for make it open for international market. So we isolate sarcomized cerevisiae from local drinks, and we standardize the production process and the tests too. So such kind of things helps us to commercialize what we have in the traditional market. This is the other projects which can be example for production of conversion of wastes to product. It is the production of briquette or energy source from sugarcane byproducts, which is bagasse, and we design the machinery, plus we optimize or we set the composition for production of briquette as energy source from sugarcane industry byproducts. This is another conversion of wastes to kind of briquette for replacement of cement from bagasse or bagasse ash, which is a byproduct from the sugar industry. These are just for you to show some exemplary researches, how what our research institute is doing so far, but these are not the only, but just for you to have highlight on what we are doing. And having said this, we can look what enabling environment available in the country for biotech. The first thing is recognize biotech as an engine for economic growth. So the government already accepted biotech as engine for economic growth, and there is high enforcement on the bioeconomy. We are drafting the first draft of bioeconomy strategy. I'm a team member of the strategic drafting group. So such kind of acceptance from the government means a lot for us because we get a lot of support from the ministries from the country related to research to biotech. The other is abundant biological resources, whether in plant, animal, or microbial, where the country is rich on different agroecology zones. So from the different agroecology zones, there is high chance of getting different microbes, plant and animals, and their interaction is very active here. There is ongoing innovative research and development programs. The research in biotech is going faster in the country. There are around 28 higher institutes, governmental institutes, giving a course related to biotech on the BSc and MSc level, and around 10 higher institutes giving at the PhD level. So there is ongoing innovative research and development programs in the country, and the expansion of higher learning institute, as I already told you, in huge number of trained use manpower, even if they are not highly, I mean, at least they have minimum skill to work on the research area, and making different products like enzyme in research laboratory at small scale. The difficulties we have is on scale up, but at the lab scale, there are a lot of starting activities on the related to production of enzymes and other research products. Growing industrial sector with growing consumer market, there is a large growth in the industrial zones. You may see on the media there are different industry zones opened within these 3-5 years. So this industry, agro industry zones and industries looking much from us, from the research area and from the market. So this enable us to work a lot. If we have the product, there are industries which are ready to receive the product. And existence of local international collaboration for biotechnology research and development. We believe that we have a good relationship with national and international collaborators that enable us to work a lot on the biotech areas. And I think we also going to collaborate with you all guys related to research activities on the biotech, and we're so happy in any kind of collaboration to enable us to scale up our research activities. And I'm so happy to share what we have in here and ready to answer if you have any question or if you need any kind of clarification on what I'm presenting. So thank you everyone for your time and attention. Thank you. I'm a second. Thank you. I'm done.
That's fantastic. Thank you, Yordi, for giving us a rundown of what goes on in Ethiopia. I'm going to just open the floor to any questions from the audiences. Is there anything that you want to bring up to Yordi? Whilst you're thinking of that, I did have a quick first question, because you said that Ethiopia the government is quite interested in doing more, investing more in biotechnology. Are there any specific sectors that the country as a whole is more interested in? Because you seem to focus a lot on sort of agriculture, food, and hollow bricks, so materials. Is that what the interest is more in?
Thank you. The higher interest is on because the country is based on the agriculture, so more interest is on the agriculture and agro-processing. And the next is the industry's need for the enzymes for processing different activities in their industries, especially the leather, textile, beverage industries are looking for the enzyme. Even the reagent industries are requesting a lot of research enzymes because they are importing with a much cost, with a hard currency. So much is on the agriculture, agro-processing, and on the enzymes for the industry sector. Yeah, that's it.
And sort of just to follow up, because you said also awareness about biotechnology and sort of the safety of using GMOs is also a challenge for you guys.
Yeah.
So how, like you said that you were doing a lot of materials to kind of in the media to get people more aware of what's being used. Do you think that's been effective? Like, do you find people are more receptive when you talk about your research?
Yeah, firstly, when you go them and tell them that you're going to explain about the biotech, they think that is all about GMO. But after you try to explain what you mean by biotech and how it is natural and how it's going to be helpful for them, they understand it differently. And nowadays, people are getting much interested on the areas of biotech, and even industries are coming to our office asking if you can produce this, if you can do this for us, if you can work with us, something like that. And even the farmers are getting knowing what we mean by biotech, because when you go to them and tell them you can produce kind of fertilizer with using such kind of bacteria, such kind of worms, and firstly they're not good enough to hear you, but when they see the product they're getting convinced, because when we go to the farmers we always go with the product. Unless and otherwise, speaking about the microbes, speaking about the strains, that's not their business. But if you go with the product of fertilizer, which is produced naturally with the bacteria, they don't have care about what strain you are using, but they want to know that's natural in this product. So if after the first visit, we pick some young people who have a better understanding what it means. So we give it training for them. We work with them on their own site, on their own land. So we produce the fertilizer together with them, and we show them easily how to produce it, and they use it on their land, and they see the product, and they're not paying anything to buy a chemical. They're just using their natural environment. So next time you go, they're ready to hear what you are going to say. We're not directly saying that we BT cotton or something like GM. We're not going to say that. Yeah, we try to convince them in the way they can understand.
Yeah, I think that definitely that show not tell kind of approach is something that we could definitely also learn from you guys. So it'll be great to talk. Yeah, it'll be great.
We spend a lot of time with them. We spend around maybe 15 days or more than that with them on seeing how they're processing their food. The one that I show you on the inset, we spend days with the ladies processing their food, and the next time we produce the food with them and we show them how the taste is, goes ours and theirs. As far as it's as their home, they trust you. That's what we bring.
That's fantastic. Yeah. Definitely love to hear more about that. But I'm going to pass on to Hanif, who has a question for Yordi.
Okay.
Thank you so much. Thank you so much, Jordan. Very nice presentation and talk regarding the biotechnological application and research in Ethiopia. My question regarding the large scale production of industrial enzyme, what you're talking about, the phytase, amylase, and some sort of xylanase as well. So what challenges currently you're facing in terms of large scale production? You are facing for the industrial collaboration, some sort of, did you target some industries in your area that currently utilizing these enzymes and their industrial preparation or not? So did you check there any feasibility of these enzyme in industries?
Okay, Hanif, thank you for your question. The problem that we are facing related to the scale up is that we don't have a large scale bioreactor. The first thing is absence of the bioreactor, and even the costs for the reagents when you scale it up, the reagents or the substrate that you are using for enzyme production is a bit expensive for us, so we can't scale it up. But related to the industries are not interested on the process. What they want is you to produce the enzyme and to sell them with the country price. Maybe if we are once successful on the production of this enzyme for the first time, the next will be easy. And related to the substrate cost, we're trying to substitute the substrate with the local wastes and different kind of substrate we are trying, but the major problem is on the scale up is case in the bioreactor.
Yeah,
Maybe,
I
maybe
currently work.
we're okay.
I myself is working also these enzymes. So I work on fructanases, and currently I'm doing on lipase and xalanase as well. So my major target is the agriculture. So it's very easy to produce these type of enzyme in agriculture waste. So I'm lucky enough to get a bioreactor recently in my project. So I'm just doing some sort of a scale up of production.
So you may be my first collaborator then in the enzyme sector. And the other gap we have is that, you see, after isolation, you need skills on the assay and quantifying their efficiency. We are simply following the normal trend. So if we have some kind of hands-on training on the production process, on the assay, it can be virtual, or if someone comes here, or if we go to the areas where we can find, we're looking also such kind of training. We're good on finding training on the molecular area, but on the enzyme area, still there's a gap.
Yeah, hopefully see you somewhere in this exploration of enzyme.
Yeah. Thank you.
And I believe I don't know if Aaron wants us to mention more, but his team in the Philippines, they're also working on the local production of purifying materials, and also working on building in-house bioreactors and PLCs. So maybe that'll be a good person to get in touch with. Oh, yeah, Aaron.
Yeah, actually, yeah. So actually, I just want first to introduce our team. So in our team, we have actually 4 kind of subgroups. So my team is the gene to protein small scale production part, and the other sub teams they design the local bioreactors and purification materials. So we are currently developing it. There is some kind of success already, and actually they have formed a kind of a subgroup here, the open bioreactor. So if you want to join in that team to exchange notes, feel free to join it, and you can contact me also if ever. So they are making we are making the bioreactors DIY, so directly from scratch using local materials. So yeah, I think that's it. And then for the purification materials, we are functionalizing the leads for it also. So yeah, we can collaborate if ever. Yeah, that's it for me.
Yeah, I just put a web link to Aaron's group in the web chat, but I'm sure, Aaron, you can provide your contact details to Yordi, so you guys can chat a bit more.
Yeah, thank you, Ella.
Lucira, you had a question. Would you like to come on chat to ask the question?
Hi, sorry. First, thanks for the talk. I would like to just ask if at what volume do you refer when you talk about the large scale of your productions, of your cell culture? Because in our lab, we work with 1 liter, and well, that's so far has been okay with us, but it's because we also don't have bioreactors yet.
Okay. In our lab, we don't have any reactor, at any volume. We are just working on a flask. That's why I'm asking, even if having the 1 liter, that will be fine. But we are on the flask range. Thank you.
Great, thank you. We as well work with that.
I mean, I think last month we did have oh, Aaron, you go ahead.
Sure, I also have another question. It's more about the natural products research of your institute. So because it's typical on developing countries to have this research, of course, because it is the developing countries who have usually have the untapped wildlife or so. But then because the developing countries doesn't have the agency to fully push on the drug discovery or product commercialization up until the FDA approval or so, usually they end up just being utilized by the first world countries. So does your country like have um, some programs or agency to ensure that these natural products that you will be discovering would be catched by a local industry?
As you said, our for the natural products, you can't get licensed, but you can license the process. But on the discovery, you can't be licensed for natural microbes. No, but you can license your process. Any other question?
I was just going to say that last month we held kind of a meeting between members of the Gathering for Open Source Hardware and Reclone, kind of a cross community meeting where members from Aaron's group were inviting people to talk about open bioreactors and other such hardware. So maybe that would also be of interest to you, Yordi and Lucira. I'll see if that could help with your scaling up of using bioreactors. So hopefully we'll hear a bit more about that. But yeah, just to echo Yordi again, are there any more questions for her? So if not, then I'd like to thank Yordi again for giving your time to tell us a bit more about all the good research that you guys do in Ethiopia, and hopefully we can connect up and hear more about the enzymes you're producing, the training materials, and how everything is developing with research in Ethiopia. From there then, I just want to go back to some updates about what's been going on in the Reclone community. So we shared earlier on a map of the world. So what we were interested in is finding out is where everyone is within the Reclone community, where you come from, and kind of in the world, where basically where our community members are, what we want within, like what we want when we come to our Reclone meetings, like what do we want to hear more about various research in different countries? Are we specifically interested in, protein expression of, polymerases and kind of other basic molecular biology tools? So do feel free to have a look at the Padlet link that I've just put into chat. Put a bit about yourselves on the map. So it'll be great to see, where we get clusterings of people. Maybe we can start building these connections, so for example, Aaron, you could put your details on so that Yordi could contact, etc. So I'll give people a few minutes to just have a look through on that. Yordi, would you mind just stop sharing screen for a sec? So we can just see the rest of the...
Sorry, what did you say?
Do you mind just stop sharing screen?
Okay.
No worries. Thank you again.
Now it stops.
Perfect. So we can see more friendly faces on screen. So as I said, it'll be great to hear, to see more of you guys and add where you are on the world. The other bit of updates. Sorry, let me just get up my notes. The other bit of update that I want to share with you guys is that last month we had Jenny talking to us about the open enzyme collection and Scott talking to us about the open yeast collection, because we mentioned that we had 20,000 pounds to spend on DNA synthesis that we can add to the kind of the research, the reclone research and diagnostic collections. So just the update is that we now have a bit more time. So if you guys had some ideas of what else you want to add to that kind of toolkit extension wish list, we have to the end of April. So I've just shared the spreadsheet there as well. If you have any other kind of enzymes that you definitely open enzymes that you want to get synthesized. The plan is that in May, we will review what's on that wish list and then start kind of prioritizing what we'll send off for synthesis. And those are the main two updates that I have from our end. Is there anything that anyone around the room wants to share about what they've been doing recently? If not, I may have to pick on people.
Well, you know what I'm doing recently? We're busy I'm doing the proposal about nanobodies, protein engineering nanobodies. It already had a good initial review by the funding agency. They advise us to consult oncologists tomorrow. So hopefully it will pass and we will have more work in here.
More work, but more exciting stuff to get involved with. And I see there's someone in the room who's called Nanobody Biotech, Romina Russi. Is this?
Oh,
or is that also? Maybe Romina has stepped away for a moment.
It's not here.
But perhaps I've got someone to also connect up with later, Aaron. We're building a lot of connections for you today. Anyone else have anything they'd like to share? If not, I won't keep you guys for too much longer if there's not much else. It'll be good to just hear more from the community. I was just taking a quick gander at the members map just to see if there's any more. We have a couple more pinpoints on it, but we'll share this around to see, because it would be nice to see who else is in the recon community and what we can bring to help each other. If there aren't any more final remarks, because I don't want to pick on people, and I'm conscious that you guys all have other busy lives and research to get back to. So no volunteers. Cool. Okay. Well, guess what then? I will just wrap up here. I want us to thank Yordi again. It's been great to hear from you. And I don't know. Have you been to many reclone meetings before, Yordi? No, just this one. Well, hopefully this has been a good start to seeing what reclone is like. And if you haven't, we'll probably send you the link to join the forum. I could pull that up now, so that we can also get in contact with you if anyone has any other ideas. So that's the forum link here. So I guess I will thank everyone for showing up today, and hopefully we'll see you again next month. It will be on the 2nd. We usually hold Rubicon community meetings on the 2nd Wednesday of the month at 1 p.m. UTC time now. So look out for our emails about the next event coming up. Thanks everyone for joining, and I guess I'll see you next month then. Take care now.
Thank you.
Bye.
Thank you.
Thanks.
Thank you, everyone. Good to meet you all. We'll
Thank
meet
you.
next month. Okay, bye.